Hello everyone, welcome back. Today we are going to learn about the second event occurring in the post-fertilization event. In the previous class we had learnt about the development of endosperm. Now this one here is the second stage where we are going to observe the development of embryo. Now we know very well that embryo can be of two different kinds. It can be the dicot embryo or the monocot embryo. Dicot, which has two cotyledons, monocot, which has just one cotyledon. But according to your syllabus, we only have this dicot embryo to be studied. So we'll be learning about that. The precursor cell which helps in the formation of embryo is zygote. So it begins with zygote undergoing mitotic division. So when zygote undergoes mitosis, it gives rise to two cells. The upper one here is called as embryo cell and the one present below is called as suspensor cell. So now we have two cells here, the embryo cell and the suspensor cell. Now this further continuously undergoes division, that is of mitotic nature. Now as a result of that, the upper one here you see embryo cell enlarges and prepares itself for the formation of proembryo. The cell called as the suspensor, which is present to the lower part, that will further undergo division, giving rise to a chain of cells. This chain of cells will be called as the suspensor. Now remember the role of suspensor. Suspensor is a structure which is meant for suspending the embryo within what? It is the endosperm in which it has to be suspended. It is because the embryo has to derive its nourishment. Nutrition has to be taken from the endosperm. So suspensor is a chain of cells formed here. Now next, here we see a globular shaped stage. This stage is seen when the embryonic cell undergoes multiple mitotic divisions, giving rise to a structure which is almost globular in appearance, and that structure is called as proembryo. On the other hand, we see this chain of cells, like I said, this is called as suspensor. The suspensor cell helps in suspending the embryo within the endosperm. The last one which we see here of the suspensor is now called as the basal cell. As this keeps on dividing, we know very well this is a dicot embryo, and so two cotyledons have to be prepared. These two cotyledons will be obtained from the proembryo. The proembryo keeps on dividing and it forms two lobes that is seen in a heart-shaped stage. So heart-shaped stage is when you start observing two different lobes obtained from the proembryo. We again have an intact suspensor, and at the base there is the basal cell. The same continues further, giving more of the appearance like seed leaf, that is the cotyledon. When we observe lastly, we'll obtain a mature dicot embryo. There's a very important component seen in the embryo. Embryo not only consists of cotyledon, but it also contains the embryonal axis. The embryonal axis contains of few regions. The first is plumule. Plumule is that part which is going to form the embryonic shoot. When the germination of a seed occurs, the first region which erupts from the seed is called as the radicle, that is the embryonic root. Followed by that, you observe another structure coming up. That structure is the embryonic shoot, which we call as plumule. So these are the components of the embryonal axis. Like we see here, the first is what? Plumule, followed by the region which is present right above the cotyledons level. We call it as epicotyle. Then comes the hypocotyle. Hypo, which stands for below. The region present below cotyledon is called as hypocotyle. The region present above the level of cotyledon is called as epicotyle. Then lastly comes this region, that is the radicle. Radicle is also known as the embryonic root. It is the first structure which emerges after the process of germination. That means during germination, the white colored portion which emerges from the seed is nothing else but the radicle. And lastly, here the radicle is protected by the root cap. So this is the structure of the mature dicot embryo. Remember these few stages. Firstly, it begins with unicellular zygote. It further divides, two cell stages seen, then it enters into a pro-embryonic stage followed by globular shaped stage, and then comes the heart shaped stage. Each lobes of the heart represent the cotyledon. In a mature dicot embryo, you will not only observe these two cotyledons, but other than that, the four regions which form the embryonal axis. The embryonal axis consists of the plumule, the epicotyle, the hypocotyle, and lastly the radicle. The radicle is also enclosed within the root cap. So this is all about the development of dicot embryo. In our next class, we'll be learning about the formation of seed as well as formation of fruit.